Madam, good morning. Hearty welcome. Today's eminent resource person, Dr. A.RAJANI Madam Garu, Assistant Professor, Head Department of Botany, Food and Nutrition, RBVRR Women's College, Autonomous, Hyderabad. Madam is going to explain one of the important topic regarding of today's class, that is CHARA. Okay, in CHLOROPHYCEAE, algae member, in CHLOROPHYCEAE, one of the important topic that is CHARA, Madam is going to explain. So my dear friends, listen carefully. If you have any doubt, please ask our respected Rajani Madam. Madam, please start your session. Please share your screen.
Okay, sir. One second. Sir, are you able to my screen, sir?
Madam, please click on slideshow.
Okay. Okay, sir.
Yes, madam. It is clearly visible. Please proceed.
Yes. Good morning, students. Today we will discuss about another chlorophyceae, last chlorophyceae member, CHARA. In the last two classes, 3 type studies are there in chlorophyceae. The first one is completed to the wallbox. The second one is Edogonium. The third type study name is called the CHARA. Here, in this CHARA, before CHARA, what is the difference in between two topics of wallbox, Edogonium, and CHARA? What is the wallbox is called the roll, to roll. The plant body is called the to roll. Next one is the Edogonium. What is the thallus? Filamentous body. The third type study of chlorophyceae, here the thallus, but you can observe to the two wallbox and Edogonium is two microorganisms. You can't see that one when I can observe the under electron microscope only. But here in the CHARA, this is the macroscopic algae. Two are microscopic, wallbox and Edogonium. The third one in the chlorophyceae, the class of chlorophyceae, this is the macroscopic algae. Okay, you can observe on direct eye contact. This is the main important character. And what is the name of CHARA? Stonewarts. Each and every one I'll explain now. Wallbox is the to roll or to round. The Edogonium is the filamentous body, and swelling bodies or swelling reproductive bodies are present in Edogonium characteristic. Here, the last topic in the chlorophyceae member CHARA is called the stonewort. What is the classification of CHARA? Same, class is the chlorophyceae, order Corales, family Coraceae, and genus name is called CHARA. Why this name is the bird is a stonewort? Why the brittlewort? Before I'll explain, it is a submerged aquatic algae. CHARA also submerged aquatic algae. The total 113 species are identified by the scientists. They are found in water bodies, freshwater, ponds, pools, lakes, streams, and attached to the substratum. How to attach it? Here rhizoids are present in the CHARA, the bottom, branched rhizoids are there. In that rhizoids to attach the substratum. Okay. Next, another one, why the name is called stonewort or brittlewort. Here, impregnated with calcium carbonate. And due to this impregnation, the plants are very rough and brittle to touch. You can observe, in the before the algae, I'll explain the cell wall to made up of cellulose. But here in the CHARA, the total plant body or cell wall to touch impaired to made up of the calcium carbonates. That's why it is impregnated with calcium carbonate, and due to this impregnation in plants, you can touch that plants very rough or brittle to touch. Hence, it is called stoneworts or brittle words. Very important. The name which algae to cell wall to made up of calcium carbonates are the stonewort or brittle wort CHARA. The names are most important. CHARA is stonewort one, brittle wort. The made up of the cell wall is the calcium carbonates. Next one coming this one. CHARA is worldwide distribution. The best example, by had it, this example is most important. CHARA nuda, CHARA vulgaris, CHARA vandalensis, or CHARA jailanthia. These three examples are most important. And that another most important in this CHARA, it is the eutrophicated and phosphorus rich water bodies. Where the eutrophication are there, where the phosphorus rich in water bodies there, you can not observe to the found in CHARA. Very, very most important. Eutrophicated and phosphate with the water rich bodies, you can't see the CHARA. CHARA is not found. This one also too most important. And the next one coming to the plant structure. These CHARA, I'll explain freshwater, green algae, submerged water ponds, and these are CHARA blatica growing in brackish water. Fragile is found in hot water streams. It is found in mostly fresh, some are freshly. These are the things I'll explain. No? In India, our India, you can observe only 30 species, which common India species. These three. These are Gelenica, Baruni, Gracillus, Hatia. These are the Indian species of the CHARA. What is the, because here you can observe the plant body, well differentiated plant body. Plant body meaning thallus. The main important of CHARA plant body is called the thallus. But here, well differentiated plant body is there. Into what is the differentiated? In the total differentiated plant body, can observe here main important rhizoids. In the substratum, you can observe the rhizoids, stem, and leaf-like structures. Not leaves, leaf-like structures are there. And another most important, highly developed sex organs and conspicuous fertilization stages are present. That's where the CHARA is known to be advanced type. Compared to other chlorophyceae member, CHARA is the advanced. What is the thallus? You can observe that this thallus, plants, you can observe the plant body, plant structure. This one is the macroscopic. The height of plants about to the 15 to 30 centimeter in height. It is made up of an erect main axis. This one, erect main axis. 1 second, see here. This one is the CHARA. Erect main axis are there. This one. And each and every axis, nodes and internodes present. And here to attach the substratum of CHARA, to attach the substratum by the rhizoid. But here, branched type of rhizoids are present. Rhizoids are multicellular. See here. These are the rhizoids. See, this is a rhizoidal plate and branched rhizoids are there. In the rhizoids are multicellular and oblique septa are present. See, this is the rhizoids, multicellular rhizoids, oblique septa also present in that one. Here, in this one, main axis, the total plant body, main axis is there, no? In that main axis, nodes and internode part and the substratum, rhizoids are there, multicellular rhizoids, obligate septa are present. Next, coming to the main axis. These main axis, two types, divided into two types. One is branches of unlimited growth. One is the branches of unlimited growth. See, this is a total CHARA picture. One, this is the main axis. See, this one is the main axis. This is a node. But here node part, this one node. In between node is equal to internode. Node, internode, main axis. This is the primary branches are lateral axis. And here you can observe the rhizoids. This is the total external feature of the CHARA. In this one, you know, rhizoids is the multicellular, oblique septa is there. What is the purpose of rhizoid? To attach to the any substrata. See, you can observe the main axis. See, this one is the main axis, this part. The main axis has two types of branches are there. One, branches of unlimited growth. Which one is branches of unlimited? This part. This part is called branches of unlimited growth. The second branch, limited growth. This one is called the second branches. Here where my arrow mark is there. This part is called the second branches. These are the limited growth. This one is the main axis is the unlimited growth, and second branches limited growth are there. First one, in this one, what is the branches limited growth? See, here nodes, internodes, I'll already explain, no? And nodal part, this part is the internode, this is the node part. In between two nodes, this is the internodal part. Internodal part and nodal part, here it's a three-layered cells are present surrounding the internodal cells species, that is surrounded by one cell thick layer called cortex. These such species are called corticate species. The cortical layer is absent in case samalge e-corticate species. Where the node part is there, no, in that node consists of two pair of central very small cells. The cells are called peripheral cells. All the node part central 6 to 21, 2, 3 central cells are there. In that central name is called peripheral centrals. Okay, coming to the main axis. The branches of the main axis to divided two branches, no? Branches of unlimited growth. See this one. And second one, branches of limited growth. First, I'll explain to the what is the branches of limited growth. These branches shows identify growth and are developed from the base of the axis of primary laterals. Base of the develop of this one. This branch shows identify growth and are developed from the base of the base of the axis of primary laterals. These function as called main branches or main axis. Okay, here limited growth, the row of 6 to 20 peripheral cells are there, where the node main axis to there, in that one branch is called unlimited growth. These are also called bracket. Branches is the primary laterals or leaves. These branches stop to grow after forming of 5 to 15 nodes, and hence it is called branches of limited growth. Next one, the second one is the branches of this is second part, this part, branches of limited growth. In this one, they are also referred as leaves, or this one is the primary laterals, or another one branches of first order. First one is the see here, branches of unlimited growth. This one is the branches of unlimited growth. These branches shows identify growth developed from the base of the axis primary laterals to produce the unlimited growth. Function is the main axis. And the way the unlimited growth of branches is there, these are referred as leaves. This one limited growth is the leaves or primary laterals or branches of first order. They are formed the row of 3 to 15 arranged on nodes. How many nodes are there? 3 to 15 or 5 to 15. That's where these branches cases to function attain particular length. That's where this is called branches of limited growth. This part, branches of limited growth. Each branch also bears nodes and internodes. You can observe the students in the unlimited branch also found in nodes and internodes, and limited growth of branches also to found to the nodes and internodes. Each branch nodes and internodes one node of each branch. Listen carefully here. One node of each branch, sex organs and secondary laterals. Here, what are the secondary laterals? Stipules, bracts or stipulates are developed the limited growth. Very, very important. Second, which branch to produce the sex organs and secondary laterals? Only branches of limited growth. This one in the middle one. Branches of secondary growth to produce the sex organs, secondary laterals like stipules, bracts or stipulates. These are developed. And what is the axillary branch? Axillary branches, unlimited growth. This one is called unlimited growth or axillary branches. Both names are same. Arise from the axis, branches of limited growth, hence it is called axillary branches or long laterals. These are also differentiated into nodes and internodes. Nodes, they are bare primary lateral. Where the node part is there, that is the bare primary laterals, and these branches look like main axis. Their growth is also unlimited like main axis. These are, I can understand, these are the branches. What is the main axis and what is the branches are there. Another one, here secondary lateral, I'll explain now in the limited growth. What are the secondary laterals? What is the stipulates? What is the bracts? Basal node, where the limited growth branches are there, we can observe to the stipulates. Where the node part is there, in that node part, limited growth developed short is there. That outgrowth is called stipulates. Sometimes these stipules, number of stipulates each node to produce the twice number. That's why that is the bistipulates. Next one, cortex region. Here many species of CHARA also. Main example, CHARA aspera, CHARA informa. Internodal cells to main axis ensheathed by the cortex region. These such species name is called corticated speech. Why? In the internodal part, cortex region sheeted is present, cells are present. That's why this species corticated species. And here, the cortex consists of vertical, elongated, narrow cells are present. Where the internodal part of it is the length by corticating filament. Develop the upper node, that is called descending layer of internode. These are the some plants ascending or descending the filaments to produce the internodes, and that is called E-corticated. Coming to the what is the cell structure? Very important in the cell structure. Why? The nodal cell containing single nucleus. The nodal cells here, two cells you can observe. The total plant body, nodes and internodes, multicellular plant body. First, you can the nodal cell, observe the nodal cell, and here nodal cell containing single nucleus. Very single nucleus, few discoid chloroplasts without pyrenoids and dense cytoplasm. Next to these cells are dense cytoplasm, uninucleate, few small ellipsoidal chloroplast is present. Vacuole is not developed inside, many very small vacuoles may be present, the nodal part. Cytoplasm can be differentiated, exoplasm and inner plasm. This is the nodal cell structure. Coming to the second one, internodal structure. Internodal cells is a very long with a large central vacuoles are present, several nuclei to compare with the nodal and internodal cells. In several nuclei, many discoid chloroplasts in well-defined longitudinal series are present. Cytoplasm shows two distinct areas. One is the exoplasm. Exoplasm is catenary exoplasm. Endoplasm is called rotating. Both are present. And streaming of cytoplasm, that is the cyclosis, is observed in this part. Streaming of the cytoplasm, that is the cyclosis. This is due to alternate contraction and expansion of the protein fibers also present fixed in the each and every cell wall. The cell wall is made up of, see here, very important, another one. Cell wall is made up of pectins, calcium carbonates. Cellulose is common, but here the cell wall made up of pectins and calcium carbonates. That's where each internode cell ensheathed by vertical corticals, and these calcium carbonates are present. That's where the plant body to touch very rough, brittle, and stone. That's where the plant name is called brittle bark. Okay, coming to the next point, reproduction. Here, CHARA reproduction in this time very important. Many times to ask the question in the reproduction, vegetative reproduction. Which algae vegetative reproduction? Amylum starches are there. Bulbils, amorphous bulbils, tubers, secondary protonema. To compare to the Volvox, Pseudogonium, the well advanced type of vegetative reproduction you can observe. That's why to focus the questions here only, vegetative reproduction type. What is the CHARA multiplied by the bulbils, amylum starch, protonema, the amorphous bulbils, tubers. First one, what is the bulbils? Bulbils, these are small rounded tuberous structure produced at the lower node and on rhizoid side also. When detach the bulbils, develop into a new CHARA plant. Example is most important, CHARA aspora. It can ask in CHARA many species to ask which species to found the bulbils of vegetative reproduction. That is the CHARA aspora. Coming to the second, very important this one also, amylum starch. This is the star-shaped cells. Many times to ask this question. Which algae vegetative reproduction to amylum starch? That's the CHARA, but species also most important. The star-shaped cells filled with the starch grain. The star-shaped cells filled with the starch grains are formed on lower node of branches, then separated from new plant to produce. But the example here, CHARA staginaria. Stagiaria is the main important. This is the amylum starch to produce the new CHARA plant. Next, coming to the next, amorphous bulbils. Here, amorphous bulb is group of many cells. Group of many cells, irregular in shape, with developed lower node part on the main axis. Example, CHARA delicatula, CHARA rhizoids, CHARA fragifera, and CHARA batylica. These are the examples for amorphous bulbils. You can observe here when the unfavorable conditions are there, these bulbils survive and make CHARA plant on retain of the when favorable conditions are there. This is the amorphous bulbil. Coming to the last one, secondary protonema. Very important this one also. Only CHARA you can observe the vegetative reproduction type of secondary protonema, amylum starch. Both are the most important. One is the amylum starch, the amylum starch, and the secondary protonema. Secondary protonema, they are developed from the node part. These outgrowths develop into a new plant when we attach the main. Here, tubular or filamentous structure are there. Develops primary protonema or the basal cell of rhizoid. These secondary protonema, just like the primary protonema from CHARA plant. These all you can observe these four, you can byhead it, vegetative reproduction. But many times to ask the amylum starch, secondary protonema, important. Coming to the next one, sexual reproduction. That's here, asexual reproduction totally absent. Only you can read out to the sexual reproduction. Very important this one also. CHARA shows a highly advanced type of oogamy type. You know, what is the sexual oogamous, isogamous, physiological isogamous, anisogamous, but here you can study it in the CHARA, highly advanced type of oogamy. Another very important, sex organs names are most important. Macroscopic sex organs. Why? The plant body also macroscopic. That's where the sex organs also macroscopic. The most important CHARA species. Some are two sex organs are there. Male sex organ name is called globule. Very, very important. Male sex organ. This is the male sex organ name globule. Female sex organ name is called, that is the oogonia nucleus. You know, common name male is antheridia, but here the name of sex organ male is called globule. This part is globule, and female sex organ name is called nucleus. Oogonia is common, but the name here nucleus. The globule you can observe here. Globule is a spherical shape and reddish color, while the nucleus is oval shape and enveloped green sheath. Spiral threads also present, but both sex organs are born on the nodes or leaves. And most of the algae species, sorry, CHARA species, monoecious, that is the homothalys. Monoecious meaning homothallic. Only few are the dioecious, that is called the heterothallic. Monothallic, best example, Kala gilanica. Heterothallic, the best example, Kala velvici. Here the examples are most important. Names. Male reproductive organ name is called globule. Female reproductive organ name is called nucleus. But here monoecious and dioecious. Homothallic and heterothallic also important. This is the examples are most important. But you can observe the former protandus in nature. The globule, see here in the picture, the globule develop below the nucleus. This is a nucleus. Always globule develop below the nucleus in monoecious species. Coming to the what is the structure of globule, how to develop the globule. Here it is a spherical shape, reddish in color. See this one. This is the globule. This one is the nucleus where the node part is there. In that node part to produce the globule. And this one is the spherical shape. Large spherical shape, reddish in color, and sometimes yellow color also present. And consist of, here see, you can observe the picture, you can observe this picture. This is the globule, spherical, long, reddish oblique, yellowish color. It consists of pedicel. See, you can observe this part. This part is called pedicel. Another name is called stalk. Pedicel or this one is called the stalk. 1 second. Yeah. See, this part is called the pedicel part. And what is the name? Another name is called stalk. And the next one, the second one, a shield made up of 8 plate-like structure. A shield made up of the shield. This one is a shield cell. The second one, coming to the second point, a mature globule is made up of curved shield cells. How many cells are there? 8. See, 1, 2, 3, 4, 5, 6, 7, and 8. How many shield cells are there? 8 plate-like shield cells. Curved 8 plate-like shield cells are there. The shield cells are filled with red color or yellow color pigment. That's where the characteristic feature of color of the antheridium, so that is the globule color. After 8 sheet of shield cell, again to the second cells is called manubrial cells. What is the second cells? No? Manubrial cells. Here, manubrial cells, see this one. This is the manubrium. Here you can observe 1, 2, 3, 4, and again backside again to 4 manubrium. Total sealed cells are the next one 8 manubrial cells is called, that name is called handle cell. How many are manubrial are there? All are the manubrial also 8. This cell another name is called manubrial cells or handle cell. Each these are arise from the shield cell. See, this one shield cell is there, arise from one manubrium. Again here shield cell. Next one is this arise from the manubrium. Like this to arrange the manubrium. Total 8 shield cell, 8 manubrial cell. In between cavities of the manubrium, you can observe the in between manubrium here 1, 2, 3, 4. In between manubrium cells, cavities of the manubrium cells, cavities are present. Cavities of manubrium, these are called, in between the manubrium, cavities are present. That cavity name is called primary capitulum. See here, this is the primary capitulum. In the primary capitulum, here you can observe inside. This is called the secondary capitulum cell. And long clip-like coiled antheridial filaments also present. These are the clip-like antheridial filament. The total output of sperms is about here 20,000 to 50,000 sperms to output. And the here biflagellate anthrozoids also present in each and every one. And main important of global structure, stack is pedicel is present, 8 shield cell arise from the 8 manubrial cells. In between manubrium, primary capitulum, inside secondary capitulum, and the clip-like antheridial filaments are present. And the how to develop in this one globule develop from the globule initial cell. How to develop globule initial cell which divide transfer division into 2 cells. One is the basal cell, second one is called the basal cell does not divide further and form pedicel. The basal cell to form to the pedicel. The second one is the upper. The first in globule initial divide to transfer division to form 2 cells. One is the basal cell. Again, second one is the upper cell. Always basal cell does not divide the further division. That basal cell to form to the pedicel. The upper cell initial divides vertical twice to form an octan. What is each cell octan undergoes? To pericytial division to form the series of 3 cells. What is the series of 3 cells? The outer cell is called shield cell. Middle cell is called the manubrial cell. And the inner is the primary capitulum. And secondary is called the bifurcate 2 cells. That is the secondary capitulum. And anthrozoidal, that is the spermatozoid filament or antheridial filament. Each spermatogenous filament consists of nearly 200 cells. From each cell, bifurcate coiled anthrozoid is formed when mature. The escape from the antheridial wall through a pore. This is the development and structure of globule. Coming to the second one, female reproductive organ name is called nucleus. And here nucleus is, you can observe this part, very large in size to compare to the globule. Nucleus is female reproductive organ. It is a large oval structure covered with green colored spirally coiled. See, this one is spirals are there. Spirally coiled tube cells forming a sterile sheath. The sheath provides protection of the nucleus. How to develop this nucleus? The nucleus develop from peripheral initial. The nucleus initial. The initial cell which divide to form row of 3 cells. Here also to form to the row of 3 cells. The basal, see here, where the my arrow mark is there. The basal cell does not divide further and form pedicel. The upper cell, this one, 2 cells initial to divide it to form 2 cells. One basal, one upper. Basal cell does not divide to form to the pedicel. The upper cell divide, the upper cell form the oogonium. The middle cells by vertical division. The middle cells by vertical division result in a 5 sheath initials encircling the oogonial. Here you can observe the 5 sheath cells are present, encircling the oogonial. These cells by the transfer division gives rise to 5 upper corona cells. See here, 1, 2, 3, backside 2 are there. Total, how many corona cells are there? Cone-like cells. That's why the name is called corona cells. 5 lower cells. You can observe lower. Tube cells. These are the 5 tube cells. The corona cells form a cap over the oogonia. When the nucleus mature, the tube cells below the corona rupture to form pores. This is the development of nucleus. See here, corona cell, how to the fertilization, how to after development of globule and nucleus, what is the fertilization? In this one, the biflagellated, see here, biflagellated anthrozoids enter into the oogonium through the pores formed near the corona cells. This is the corona cell. A single anthrozoid fuse with the egg to form a diploid zygote. This one to form the diploid zygote. The zygote create a very thick wall. Why the protection? That was a very thick wall. The sheath cells disintegrate, the zygote undergoes a period of rest. After rest, you can observe this one. After rest period, the germination to zygote. Before germination, here which the nucleus is there in the zygote? Diploid nucleus. The diploid nucleus migrate to the anterior region where it is the meiosis is there, that is a haploid. And these cells, 3 nuclei degenerated, only 1 cell is formed, that is the rhizoidal initial, that is the protonema is formed. This divided into the nodal part, internodes. The second node of the